All right, everyone, we're going to do a virtual tour today, July 12th, 2017, kind of looking at some of the ecohydrology work as it maps onto the hyperspectral and multispectral imaging. We have Dana Chadwick on the right from Stanford University, Nicola Falco from Berkeley Lab. So why don't you guys tell us what you're doing here today?
All right, so we were able to do some hyperspectral imagery collection, and so now within the footprint of that collection we're setting out plots where we're doing measurements with a field spectrometer.
We have been taking like plot level measurements, and now we are taking also leaf measurements, and we also sampling those leaves for chemical analysis and bring back to LBNL for more
I see.
analysis.
So you're doing some level of census work, so inventorying the plants that are within this 1 meter by 1 meter square.
Yep, and we're also going to measure the height and do some destructive harvesting so that we can calculate the amount of biomass that's existing in these different plots along this landscape.
Excellent.
So the
And
plan
go
is
ahead.
to have at least 8 or 10 plots along this line where the image has been taken. So the diversity is quite high, and we try to get all the M members of this area.
Excellent. And so this is the handheld hyperspectral imaging
so this
device?
Is the handle, and it has a clip where we can insert the leaves. It has a lamp where we can use as a source, so we don't need any solar measurement or solar radiation. So we are totally independent by the weather conditions this way.
Excellent. That makes life a lot easier. Just to put things into perspective, one of the issues we had with the hyperspectral overflight on the drone was that at this elevation, the drone didn't have quite as much lift as was expected, particularly toward the end of its flight. So it came in for something of a hard landing and damaged the camera. So the transect that we're sampling along, for which we have hyperspectral data, is unfortunately not directly overlapping with our ecohydrology study plots, which you can see in the distance. However, we do have overlapping multispectral data, both collected by the Oregon State team as well as periodically by the Berkeley Lab team. It's actually co-located with the hyperspectral measurements, but for which we have the entire study domain covered. So hopefully it'll be a great opportunity to kind of compare those two methods and see how this all then maps actually to the plot scale. Thank you both.
Thank you.
Thank you.